first speaker of today. I'm very grateful to the great Dan Villeneuve from the US EPA that has woken up in the middle of the night to give us the first talk. Dan, if you would like to start sharing your screen. Thank you very much.
All right. Hopefully you can see that and hear me.
Yeah.
Great. So thanks for the invitation. Really enjoyed the meeting yesterday. And so today I'm going to be talking about some work that we've been doing at the US EPA. First, I want to give an introduction to kind of our first standardized transcriptomics-based chemical assessment product. So I'll give you a bit of background on that and then talk about some of our ongoing research to develop a similar type of assessment product for ecotoxicology. So within the EPA, I work in our Chemical Safety for Sustainability research program. The challenge that we're facing, very similar to what Jason O'Brien described yesterday. We have a chemical landscape that's large and growing. We know that over 95% of manufactured goods are reliant on some kind of industrial chemical process. There's over 350,000 chemicals or mixtures that are registered in one or more inventories among 19 different countries. In the US alone, our inventory under the Toxic Substances Control Act contains over 86,000 chemicals, 42,000 of which are known to be commercially active. And of course, these numbers are increasing over time. So this presents a challenge to our traditional chemical risk assessment paradigm. On the human health side, from the time of identification of a new chemical that warrants assessment to actually developing all the assay data, finalizing the report from the studies, depending on studies that are ordered, can take anywhere from 2 to 10 years to complete. Then to take those studies, perform a typical risk assessment, takes several more years. So ultimately, on average, we're talking 6 to 14 years for a human health oriented assessment under this traditional guideline whole animal testing sort of paradigm. And as a result of that, as you might expect, fewer than half chemicals that are in commerce and being used have actual traditional toxicity testing data and comprehensive data sets. And you can see here across a wide variety of inventories of chemicals that have been detected in the environment, chemicals that have been detected in the human body, for example, emerging contaminants like PFAS and industrial chemicals on our Toxic Substances Control Act inventory. A lot of these different classes, there's very little toxicity data. And so this is problematic. The time that it takes to reach decisions on these chemicals has been a challenge for our agency. A recent value of information analysis by EPA basically evaluated the importance of time as a consideration in the choice and use of toxicity testing methods. And really, when considering the choice of 2 different toxicity testing methods, one that could inform a decision sooner, but perhaps has greater uncertainty or variability associated with it, versus waiting longer, making a decision later based on more certain data. They find that there's considerable return on investment and socioeconomic benefit in terms of reduced health costs by making a decision sooner, even if that's based on somewhat less certain toxicity information. And so in thinking about how to address this in EPA's portfolio of chemical assessments, really we have a gap when it comes to an assessment product that can take a chemical for which we have little or no data and provide an assessment in a fairly short period of time. So we have things like IRIS assessments, provisional peer-reviewed toxicity values, et cetera, that are fairly data intensive, take a long time to develop, and would be considered kind of gold standard sort of assessments. but can we generate something that functions down on this other end of the spectrum where we have fairly little data and need to develop something to support decision making over a fairly short period of time? And so for this, we're really looking toward new approach methodologies, which again, Jason introduced yesterday. These are generally any technologies or approaches that can help to increase the efficiency of toxicity testing and chemical risk assessment, and at the same time help reduce our reliance on animals, reduce the overall need for animals in testing. Jason gave a really nice introduction to the concept of a transcriptomic point of departure and the possibility that we could look at transcriptomic response in animals over fairly short-term tests, whether those be rodents or potentially other organisms, and determine the concentration at which we see concerted changes in gene expression in response to that chemical and the concentration at which that occurs. Now, that activation of gene expression is not necessarily adverse, could be just reflecting an adaptive response to that stressor. But the basic concept is that if we keep the concentrations below those that actually elicit a gene expression response, we're unlikely to elicit toxicity even over much longer terms of exposure. And so we can use this transcriptomic point of departure as essentially a predicted no effect concentration. And so this was the approach that the Office of Research and Development decided to take in terms of developing a shorter term, more rapid assessment product that could provide information quickly on a data poor substance and very rapidly provide a toxicity reference value that could be used in support of decision making. So this EPA transcriptomic assessment product was put forward for a review by our Board of Scientific Counselors in 2023. There are a couple of documents that were submitted to the BOSC. One was a scientific support document that lays out the scientific rationale for this type of approach, the supporting evidence that shows that it can work, that's concordant with chronic toxicity testing. And then along with that was a standardized method for how these transcriptomic assessments would be done. So this was reviewed in July of 2023. You can find the link to the report here if you're interested in the comments, but ultimately this assessment product was approved for use within the Office of Research and Development to do these chemical assessments. The scientific support document itself consisted of a comprehensive literature review that explored the concordance between the concentrations at which chemicals disrupt gene expression and longer-term toxicity. That literature review identified over 140 chemicals tested in 32 different types of studies. The studies covered a range of different exposure routes, multiple different exposure durations, ranging from less than a day to up to 90 days, considered 8 different tissues, multiple different sequencing technologies, and chemicals with a broad range of physicochemical properties and toxicokinetic half-lives. Among the chemicals that had chronic bioassays, the transcriptomic benchmark dose was highly correlated with chronic apical benchmark doses. And the overall concordance in terms of variability, et cetera, that was found for the transcriptomic approach was in a range that was similar to that in the standard deviation of lowest observed adverse effect levels if you repeated systemic toxicity evaluations with a repeated dose toxicity test. So overall, the scientific support document showed that the concordance was robust across different exposure durations, routes, species, sexes, target tissues, etc., and showed a lot of promise to be able to use this transcriptomic point of departure to predict concentrations below which chronic toxicity would not be expected. The actual method itself for the EPA transcriptomics assessment product involves three main steps. The first step is really a comprehensive database and literature survey to verify that there are no existing guideline sorts of toxicity studies already out there. So if we have a new substance, the first thing we would do is go to EPA's ToxVal database, see if there are any established or already published guideline studies. If none are found, we would then go to the peer-reviewed literature, do a systematic evidence mapping, look to see if there are any studies that basically conform to kind of that standardized test guideline. And if those studies exist, the assessments would be based on those traditional types of studies. But in the absence of those kind of data, we would then proceed to the second phase of the transcriptomics assessment product, which is basically developing the transcriptomic data. So the standard protocol for this assay is a 5-day repeat dose study in male and female rats. They're exposed for 5 days. We then perform gene expression measurements on up to 12 different tissues. Benchmark dose analysis or dose response modeling is then performed on the gene expression data that come from those 12 different tissues. And a transcriptomic point of departure is defined as the dose at which there's no coordinated transcriptional change that would indicate, again, not necessarily an adverse effect, but potential for some kind of chronic toxicity at concentrations that exceed that. So basically, at the end of this experiment, we've got a transcriptomic point of departure. The final step in this process is to then take that transcriptomic point of departure determined in the rat model, convert that to an equivalent dose in humans based on the allometric scaling, and then apply a standard set of uncertainty factors that account for that rodent to human kind of extrapolation. And ultimately, once those uncertainty factors and allometric scaling are applied, we derive a transcriptomic-based reference value that serves as an estimate of the daily oral dose that is likely to be without appreciable risk of non-cancer health effects over a lifetime of exposure in the organism. And that data is then reported in a standardized assessment template. The reporting, review, and release of the EPA transcriptomic assessment product is also one of the ways that we really gain efficiency with this product. So typically for chemical assessments within EPA, an assessment is developed for a chemical, and then that assessment, that chemical-specific assessment, is subjected to peer review, comment, revision, etc. afterward. The way this transcriptomics assessment product works is that the standard methods for developing this transcriptomic based reference value, the standard methods themselves have been subjected to peer review. And so the methods have already gone through a Board of Scientific Council review. They've been revised according to that. So now the idea is that we generate the data using this standardized and approved method. And then the results of those data are put into a report that involves minimum free-form text, no subjective interpretation. It's just a report out of the results. That's reviewed for quality and consistency with the method, and then immediately released. So this allows for rapid experimental execution, streamlines the review process, and basically allows us to move from identification of a chemical and initiation of testing to release of the report to support decision-making within less than a year's time. And so the first actual EPA transcriptomics assessment product was performed for this perfluorinated chemical in March of 2024. So you can see in less than a year after the actual method went through the Board of Scientific Council review, we're able to take and produce the results for a novel substance and generate the report, publish that, make it accessible to support decision making. For this particular chemical, there were 9 doses tested plus control. Again, multiple tissues were evaluated in male and female rats. And the most sensitive transcriptional response in this case was in the female uterus. So the lower bound benchmark dose for the concentration at which the gene expression was affected was at 0.12 milligrams per kilogram per day. Following the allometric scaling, the human equivalent dose is shown here as 0.027 milligrams per kilogram day. Again, dividing that by the uncertainty factor, we come up with this final value that is then used in the report. In the case of this PFAS, that estimated transcriptomic-based reference value was very similar to chronic reference doses that were developed using more traditional testing for a range of other perfluorinated chemicals with similar types of properties. So this is basically the human health oriented EPA transcriptomics assessment product as it exists and is in use today. But we're here at a meeting on ecotoxicology, and ecotoxicology is my focus as well. So the question is, well, what about ecosystems? So ecological risk assessments face similar needs in terms of needing to estimate exposure concentrations that would not represent appreciable risk for adverse effects, even following chronic exposures. But we have this additional challenge of trying to protect taxonomically diverse species. We're not just concerned about humans, we're concerned about whole diversity of life. And so question we've been working on in our research group is, would an ETAP-like a transcriptomics-based approach work for ecotoxicology as well? And so we've been exploring this in our research program over the last 5 or 6 years now. We've been working through a number of different questions and issues as it relates to what a transcriptomic assessment product would look like for ecotoxicology. This has ranged from looking at what species should we test, how could we kind of give coverage of that taxonomic diversity that we're concerned with in ecotoxicology. For us, the challenge is not so much a question of how many tissues do we grab from a rat, but rather how many species do we need to represent in our testing approach in order to feel like we have some confidence in spanning that taxonomic diversity. How do we design the assay to allow for fast and efficient testing? Next question is, are those transcriptomic points of departure actually protective relative to apical endpoints? What about subapical? How do those compare? How many model organisms do we need? Obviously, we want to cover taxonomic diversity, but for efficiency of testing, we want to keep that number of species as small as possible. We also need to evaluate assay performance characteristics, and then think about, as we generate these novel data, how can we apply these in different types of decision-making contexts? So I'm just going to give a couple examples from our research to date that try to address these various questions and issues. So first of all, relative to species selection and assay design, for our ecological high-throughput transcriptomic research at EPA, we've been focusing primarily on 4 different species. So in algae, an aquatic vertebrate, so typically larval fathead minnow, but we've also tested zebrafish, crustacean, Daphnia magna, and an insect larvae, Chironomus dilutus. These 4 species basically represent genomically, physiologically, taxonomically, and trophically diverse species. In terms of the food web, it covers primary producers, primary consumers, secondary consumers. These species are also commonly used in the globally harmonized system for classification and labeling. An aquatic vertebrate, crustacean, and an algae are what's typically required there for labeling purposes. And of course, aquatic organisms are highly vulnerable to exposure. So the assay itself is intended as a high throughput assay. So we basically load organisms into a deep well 96 well plate. individual organisms in each well. In the case of the aquatic vertebrates or fish, we use larval fish at an early life stage that can fit into the wells of this 96 well plate. We then use automation to dose that plate with a concentration response curve chemical. We actually use a 3D bioprinter on our team, which allows for delivery, precise delivery of nanoliter concentrations or nanoliter volumes of chemical to those plates. We expose them for 24 hours, again, trying to keep the test short and efficient, minimizing the need for water changes, etc., over the course of the exposure. After 24 hours, we do morphological observations to evaluate survival and changes in morphology. Also collect things like photosynthetic efficiency for algae, etc. We then homogenize the whole body of the organism, collect either RNA or just a tissue homogenate, use various types of sequencing approaches, either full RNA-seq or, as John alluded to yesterday, we can also use a set of sentinel genes and a more targeted transcriptomics approach. Once we've generated our transcriptomics data, we then apply dose response modeling to each of the genes that shows some kind of dose-dependent response. We calculate a benchmark concentration at which those genes respond. We then take the distribution of those benchmark concentrations and derive a transcriptomic point of departure. In our case, usually as the 10th percentile of this distribution, that represents our gene expression altering concentration. So how do these transcriptomic points of departure compare to traditional apical endpoints? So to evaluate this, what I'm showing here in this plot, we've got 22 chemicals listed along the y-axis. These are chemicals for which we derived transcriptomic points of departure using a larval fathead minnow bioassay. The box and whisker on these plots are basically the apical effect values from information that was captured in the Ecotox knowledge base. So this represents studies done with fish that focus on survival, growth, or reproduction, but this is a wide variety of study designs, different exposure durations, different species of fish, different routes of exposure, etc. So you can see a pretty wide distribution of points of departure for these traditional tests. But overall, our transcriptomic points of departure, shown in the orange dots here, are generally well below the concentrations at which apical effects were reported. We had a few exceptions to that out of this initial 22. So 3 out of the 22 that we tested had transcriptomic points of departure that overlapped those apical concentrations. But actually in 2 of those 3 cases, once we adjusted for the actual measured concentration in the well, we only had 1 chemical for which our transcriptomic point of departure was not protective. So comparing our transcriptomic points of departure to other types of effects that are coded in the Ecotox knowledge base, things like gene expression, hormone concentrations, changes in behavior, histology, etc. What we find there is our transcriptomic points of departure are not necessarily lower than the effect concentrations at which these subapical effects occur, tend to kind of overlap that same range. And so that kind of suggests that, again, we're protective relative to these apical outcomes of survival, growth, and reproduction, but not necessarily over-conservative relative to just kind of these biological changes in response to a stressor. And so that suggests TPODs are probably conservative, but not necessarily overly conservative. So the question of how many model organisms do we need to test? We don't have a final answer for this yet, but we've certainly been exploring this question. One of the early questions that we had is, could we just use the transcriptomic point of departure for fish to be protective of all aquatic organisms? And we pretty quickly found that that would not work. So again, testing a whole series of data-rich chemicals, we found that fish-based TPODs were protective for invertebrates some of the time, but definitely not for all chemicals. So for things like estrogenic chemicals or herbicides, the fish TPODs were protective of invertebrates, but things like organophosphates and PFAS, they were not. This really emphasizes the importance of including one or more invertebrate models into an ecological transcriptomics-based screening program. We also saw this for PFAS, the importance of including a range of different taxa, testing 22 different PFAS in this transcriptomics-based approach. You can see that in general, the fish were the least sensitive to PFAS, so they're represented by these blue squares here on the plot. The invertebrates are represented by the red triangles as well as the purple circles here, and you can see that Daphnia and the chironomid sort of vary back and forth in terms of which is second most sensitive, and actually the most sensitive, the majority of the PFAS that we tested was actually the algae. So again, reflecting the importance of capturing some taxonomic diversity in an assessment product based on transcriptomics for ecology. We've been doing work to evaluate various performance characteristics of these assays. So one of the things that we've looked at, particularly when we have some additional transcriptomic resources available, or just testing the same chemical multiple times to see if we get essentially the same answer. We've tested cyanazine now up to 11 different times, and we can see here the range of transcriptomic point of departure estimates spans over about an order of magnitude, so not bad for the repeat testing of the same chemical. In the case of Maslow, which we tested twice, our TPOD varied by about a factor of 2. Aldicarb varied by about a factor of 4 when it was repeated. And in the case of methamyl, the first time it was tested, we got a negative response in the assay. And when we repeated that, we got a negative again. So showing consistency in assay responses and the TPOD estimates. Again, I mentioned earlier that we also have explored the use of sentinel gene approaches, where instead of testing the whole transcriptome, we test a smaller subset of genes that are intended to represent the overall variation of the transcriptome in response to the stressor. So in the case of Daphnia, we have a subset of 2,400 genes that represent about 5 to 10% of the overall transcriptome. So we took data from whole transcriptome studies with 18 different PFAS. We then subsetted out this smaller set of sentinel genes and derived the TPOD from the whole transcriptome versus this smaller subset. Generally, we see pretty good concordance between those. And in cases where we deviated from that concordance, in the majority of cases, we were more sensitive with the sentinel gene set than with the whole transcriptome alone. So there are very few cases, and the cases in which we had a very small number of concentration responsive genes where we'd come up with a less conservative estimate using the sentinel gene approach. So that gave us confidence that we could use this kind of reduced transcriptome approach. We've also looked at variables like experimental design. So we did a study in zebrafish where we looked at different windows of exposure during the embryo larval period. So we have, for example, exposure from 6 to 24 hours, 6 to 48 hours, 6 to 120, 24 to 120, etc. Lots of different variations. And what we see is that in most cases, we have a transcriptomic point of departure that's kind of in the same range within about a factor of 10 or so across these different PFAS that we tested. The major exception to that is exposures that were done for that 6 to 24 hour post-fertilization period that seem to be considerably more sensitive, at least for 2 of the 3 PFAS that we tested. Finally, we've been exploring the application of these data in risk-based screening for data-poor chemicals. This really evolved from work that we were doing in the Great Lakes. We have 10 years worth of Great Lakes tributary monitoring data. In that process, we've detected over 150 different contaminants of emerging concern that are detected out there in Great Lakes tributaries for which we have no other toxicity data. So we picked 10 of those data-poor chemicals and we ran them through our high throughput ecological transcriptomics assays. We did 3 different species, so fathead minnow shown in the blue triangles. The red squares are the Daphnia magna, and the green dots are the algae. And then these black triangles with the error bars down here, this is the range of environmental concentrations that we detected in Great Lakes tributaries. And so you can see here, generating these data, in most cases, those concentrations are well below those that cause gene expression changes in these organisms, suggesting there's probably pretty low risk for effects out at those environmental concentrations. If we were going to test any of these, we have a little bit of overlap here between the most extreme concentrations and the lowest range of our algae transcriptomic point of departure. So we might prioritize these two chemicals and testing in algae if we were to then move to another tier of testing. We also took our existing transcriptomic points of departure for some of the more data-rich chemicals, compared those against the environmental concentrations. And here you see we had a number of cases where the environmental concentration or maximum environmental concentration, represented here with the black dots, exceeded our fathead minnow-based transcriptomic point of departure. And so these are cases where, just based on a risk-based screening context, we might prioritize those chemicals for additional testing, or at least propose that there might be potential for effects at some of those sites based on the concentrations of these chemicals exceeding the transcriptomic point of departure. So showing how we could use this in kind of a screening and prioritization context. So overall, transcriptomic points of departure are in use already for predicting no effect concentrations pertinent to human health. This is our current EPA transcriptomic assessment product. Value of information analysis has shown the benefit of a rapid, possibly less precise determination and decision making over a slower, more precise decision making method. Our ecological high throughput transcriptomics research has shown the transcriptomic point of departure to be promising for predicting no effect concentrations for ecotoxicology. We've already applied this approach for risk-based screening and prioritization, and we're now doing the research and working toward development of a standardized method for an ecological EPA transcriptomics assessment product. And so we've got still a number of research questions to resolve, including which species, how many species do we need to provide adequate protection relative to something like species sensitivity distribution. In the case of aquatic vertebrates, does the whole body of the larval fish serve as a reasonable surrogate, or do we need to do a whole body target organ function transcriptomics in adults to really be protective? And overall, just trying to develop the scientific support for an adaptation of this ETAP approach for ecology. So with that, I just want to acknowledge there's a huge team of folks that contribute to this research and really make it all go. And so just want to acknowledge them and all their contributions to the work. And with that, be happy to take questions if we have time. Thanks.